Vision session with me, Christine. So today's lesson, I want to look at photosynthesis and photosynthetic pigments. So let's just take a step back and remind ourselves about that structure of a chloroplast. So we know that we've got this minute biconvex lens, and we know inside there we have thylakoids that stack up to form what's known as the granum. Well, what we want to do now is we want to look inside that thylakoid. And we want to look at the thylakoid membrane, because embedded in that thylakoid membrane are these photosynthetic pigments, along with those proteins, and what they form is what's known as a light harvesting system. So they are what's known as an antennae complex. And they've got a very specific role to play in that what they do is they're going to absorb the light energy that's going to come from the sun. So let's just go back to GCSE physics and remind ourselves about the visible light from the sun. So we know the visible light is composed of a range of wavelengths, and those wavelengths go from about 400 to 700 nanometers. So if we ever see a rainbow, what we're actually seeing is we're seeing the refraction of those different wavelengths, the separation out into the different colours that we see. Well, that's important for us to understand that our photosynthetic pigments actually absorb very specific wavelengths of light, and they will reflect others. So if we look to the primary pigment, chlorophyll A, they absorb mainly red and blue light, and they reflect green light. And that's why we see plant leaves as being green, because there's lots of chlorophyll which is reflecting the green back into our eyes and absorbing the other colours. Well, what happens in autumn? The amazing thing that we can actually see is we get to see in autumn time these accessory pigments, these other pigments that make up the antennae complex. Our carotenoids, our xanthophylls, our chlorophyll B. These are all known as accessory pigments, and these all absorb different wavelengths of light. And we can see that if we look at the leaves as they change colour. But how do we really know this in a scientific way? Well, what they'll want to do is link this to your practical side of the course, linking it to that thin layer chromatography. So they may give you an example of what they expect you to have done to extract some of those pigments. So in this case, they've given an information in an exam question. So there's step 1, step 2, so lots of information that's been given here. As you go through this, the first thing I would suggest that you do is highlight some key areas, because what they tend to do is ask you to analyse why might the students have done this? Why should you be doing these specific parts of a practical, and how could you improve on it? So one of the things that it says here is that you need to hold the TLC plate very carefully by the edges. You want to avoid damaging the surface. Why would we want to do that? So what they're checking here is, do you understand that we want the pigment, the movement of the pigment, the spot, to move up through the TLC, the thin layer chromatography plate, and we want to ensure that we do not affect the movement by damaging the plate. We want to ensure that it will only move from the position, the line that we draw it across. So you're using a pencil line, and what you're going to do is you are going to see that the mobile phase, that organic solvent, is going to move, and it's going to move up from the bottom up to the top. So that mobile phase, that organic solvent moving, we want it to move, and we want it to pick up the pigments and to therefore carry them in a direction which is upward. So we don't want it to move off to the side, and that's why we need to make sure we hold our TLC plate very carefully by the edges. We also want to ensure that we don't actually contaminate at all. We don't want to put any of our fingerprints on it. So we need to make sure that we're only having the movement of the pigments up with that organic solvent. But what causes them to actually stop? What causes them to separate out is to do with our stationary phase. So our thin layer chromatography has a silica gel in it, and what we want is the hydrogen bonds to be formed with the silica gel and the pigment, and the strength of those bonds are going to ensure that our pigments separate out into different layers. So what we've got is these different photosynthetic pigments are going to have these different attractions to that stationary phase. And because of that, what you're going to end up with is the separation out of those pigments. So we can actually see the chlorophyll A, the chlorophyll B, the xanthophyll, the carotenoids. Now, one of the other things they do love to do is ask you about how to work out your RF value. So you need to remember, to work out your RF value, you want to look at the distance which has been moved by the spot divided by the distance moved by the solvent. And they love giving you a question like this in the exam, and they expect you to use a ruler to measure those distances, or they expect you to interpret the results. And if they're expecting you to interpret the results, they are going to give you, for example, the picture on the left, figure 18.3, where they're saying, well, sample A actually has 2 pigments in it, whereas sample B has 3. So they want you to be able to not only calculate that RF value, but they also want you to interpret the data that they've given you. And again, they want you to actually look at the practical application. So why a pencil line across the width? Why pencil? Because the pencil will not form hydrogen bonds with the mobile phase, so it shouldn't move up with it. The other thing is also about make sure the plate does not touch the side of the jars anyway. The reason being, if it touches the side, if your plate is too wide and is going to touch the side, that's going to cause your movement to move in the wrong direction. So you could end up with it moving off to the left or off to the right, which then means you won't get a correct reading for that RF value. So that's how we can see the pigments, but let's just take a look back at these pigments and let's talk about what we actually have. Well, the antennae complex I discussed earlier, that's not the only part which is going to be involved in the absorbing of the light energy. We know our accessory pigments are going to absorb light energy, and they're going to transfer this light energy to what's known as a reaction center. Now, chlorophyll A is the primary pigment, and that is what's located in that reaction center. And what you have to understand is chlorophyll A actually becomes oxidized. And it becomes oxidized, which means it's able to donate an electron to the electron transport chain. So when we look at the accessory pigments and we look at the reaction center and we look at this whole combination of an antennae complex and a reaction center, we discuss it as being what's known as the photosystems. So our light harvesting system and our reaction center is actually called a photosystem, and there are 2 photosystems that you need to know: photosystem 1 and photosystem 2, and I'll go into more detail on their role to play when we look at the light dependent reaction. So right now, what's important for us to understand is these photosystems are made up of this antennae complex, this light harvesting system, and a reaction center. These accessory pigments are going to absorb the light energy and transfer that energy down to the reaction center. That's why we call it an antennae complex. It's funneling down to that reaction center. When we get to chlorophyll A, this is where the important parts start. We're going to have the fact that the chlorophyll is going to be oxidized because it's donating its electrons. Well, what does it mean? How is it going to donate its electrons? And this is where we have to take a step back and remind ourselves about GCSE chemistry and physics and about the atoms. So if we look at our atoms, we should know that we have our electron shells, which are a specific energy level. Now, when a photon of light is absorbed by a pigment, its energy is actually transferred to the electron which is absorbing it. So if that photon of light is going to be absorbed by that electron, this is going to excite the electron, causing the electron to jump to a higher energy level. We call that its excited state. So as the electron then moves through that electron transport chain, what's going to happen is it will then release that excess energy and it returns back down to its lower energy level. If you like this video, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.